Okay guys, I'm going to go over PCR with you. I'm going to make it quick and easy for you. In addition, I've given you an extra video to watch that animates this. So in other words, it brings it to life, okay? PCR is what you would have done had you gone to Mac. So they have the PCR machine there, and PCR is this. It's called polymerase chain reaction. You should recognize the term polymerase there. It's a chain reaction, which means it's happening over and over and over again. What is PCR used for? It's used to make a lot of DNA, okay? And the term for making a lot of DNA is amplifying. So if you want to make a particular section of DNA or an entire DNA molecule, usually they go for a particular section, they go through PCR. So in other words, if you get a little bit of DNA from a crime scene and it's not enough to put on that gel that you looked at a couple days ago, you can take it and make more and more of it so that then you can plot it on the gel and actually compare it to suspects at the crime scene. So this could become your ladder, okay? So what does PCR consist of? It consists of three very simple techniques, okay? But it wasn't so simple when it was developed, so I do want to give credit where credit is due. It was developed by Kary Mullis in 1983. Okay, and it's used all the time now. It's actually becoming a little bit outdated from what I hear at Mac, but they still have their PCR machine and they use it for their genetics labs and their first year university labs or bio labs. So polymerase chain reaction, okay, these are the three steps to making more and more DNA. You have to denature your DNA, okay? So denaturation, normally we don't think that's a very good thing. We have to take the molecule apart. All right, so you would have heard the term denature way back in Unit 1 when we talked about proteins. Okay, so the two-stranded DNA has to be separated into single strands. All right, so we're going to denature. Now, how do we denature this DNA? We have to melt it down. How do we do that? We bring it to temperatures between, depending on the machine, between 92 degrees Celsius and 96 degrees Celsius for 2 minutes. So think about that for a second. Think about your body temperature, okay? And think about other body temperatures of different organisms. That's quite high, all right? So this is Celsius, not Fahrenheit. So this is a very high temperature. So they put the DNA in very high temperatures, okay? And they denature it. Next, we anneal. So we prevented annealing with single-stranded binding proteins. Here we want primers to anneal to the DNA that we've separated. So separate the DNA in denaturation and then get some primers in there, and they're added in excess. So I wanted you to know that in excess. In other words, there's lots and lots of primers put in there. So it increases the chances of the primers binding to the DNA strands. When annealing is accomplished, the temperature has to be lowered. Okay, so it has to be lowered to 68 degrees Celsius, and they keep it at about that for about 60 seconds. So it goes from very high temperatures to separate the DNA molecule to lower temperatures, not low, but lower temperatures to allow the primers to attach to the DNA. All right, and then it says, as a result, the primers form hydrogen bonds and anneal to the DNA on both sides of the DNA sequence. And then finally, okay, we've got annealing, we've got primers. What do primers do? They tell the DNA polymerase where to copy, all right? And so then we have to get the DNA polymerase in there and start copying the DNA, and that's called extension. So finally, different nucleoside triphosphates. Nucleoside triphosphates provide energy, remember that? It's a triphosphate, and then two phosphates will be used for energy to power this machinery. So nucleoside triphosphates are added, and a thermostable polymerase, DNA polymerase, is added. So I left that blank because I like to just make sure that you guys are aware what that thermostable polymerase is and why it's of such importance. So think about this. Your body temperature is nowhere near the temperature of this machine, okay? And most organisms aren't near the temperature of this machine. So they have to be able to get an organism whose polymerase, okay, you and I have polymerase too. We know that we've got polymerase 1, 2, 3. But they have to be able to get a polymerase in there that could withstand such temperature fluctuations and such high temperatures. And so trial after trial, they found one that is a bacterial polymerase. And I just grabbed an old pen here. So the polymerase is called Taq polymerase. Taq is the species of bacteria that was able to accomplish this. So TAQ polymerase, okay? Taq polymerase. Taq is the species that allows its polymerase to go to such a high temperature without being completely annihilated, all right? So Taq polymerase was discovered, and that's the polymerase that we use now, all right? So it belongs to a bacteria species known as Taq, and there's a very long word for it. You don't need to worry about that. So Taq polymerase is added to the reaction mixture. It helps polymerization process of primers and therefore extends the primers at 68 degrees, okay? So that happens, and then the DNA is made, and then the whole thing is done over and over and over again. All right, and so they suggest that if 20 cycles occur, about 1 million copies of the DNA that they've targeted are produced. All right, that's a lot. So diagram here now, I think you can understand it. Denature, okay, take it apart. This is denaturation. All right, so this is step 1. Take the molecule apart. Add your primer, step 2, high temperature, lower temperature, but not low. And then this is stage 3 with elongation, okay? And what do you get? With one original DNA molecule, you can see you've separated, you've made 2, okay? And that's after 1 step. After 2 steps, you would get 4. Now, it's not quite that easy. And I'm going to show you, I've put up a video clip there that shows you exactly why it's not quite that straightforward. And to get you to make sure that I'm furthering your education for you, I'm going to get you to do, you'll find out in a few minutes, and it's already posted. There's a question 4 on page 53 of your booklet that I want you to illustrate. So you're going to draw this out. The video, as long as you watch it, is totally going to help you, okay? And so this is going to be a communication mark for you. But this starts you off showing you that after 1 cycle, you'll get 2. After 2 cycles, you will get 4 DNA molecules, okay? But some of these guys are going to continue to work, and some of them are going to just be products, okay? And please watch the video to find that out. All right, and lastly, how do we use PCR? Okay, forensics. Number 1, DNA fingerprinting. You can do paternity tests. That was the word I was lacking a few days ago when I tried to tell you guys about that. So paternity tests, criminal identification, diagnosis of microorganisms, right? You get a little sample of DNA, you can find out what kind of microorganism you have as long as you amplify it. In other words, one little section is not going to do much for us. We have to be able to make a lot of DNA to put on the electrophoresis and identify it according to the ladder that we already have. So if you think you've got E. coli will have certain bands, and then you can amplify your DNA and say, yep, those are the same bands. Now I can see these bands because I have a lot of DNA. How did I get a lot of DNA? I got it from PCR. Studying evolutionary biology, I gave you that example with the deers and looking at the bands. Fossils, okay, looking at paleontology, very similar, okay, but you can get DNA from fossils. Gene cloning and expression. So if you want to clone, you want to see if you've made all the DNA possible. Gene sequencing, okay, vaccine production, I'm just reading these as well as you could, and drug discovery. So these are all applications of how PCR can be used, okay, in all these different areas. And in terms of vaccine production and drug discovery, I've given you a little clip on COVID-19, okay? So I just wanted you to know that what you're learning is completely relevant to 2020, and just watch that little clip showing us how they're actually using PCR and COVID-19 testing.